causing. In some cases, it's a relatively trivial disease, just like a minor cold or a flu. In other cases, they can be fatal diseases like AIDS. But viruses, each and every virus is a disease-causing organism. And they, as I said, the virus themselves are so simple they can't reproduce themselves, but they have to get inside a host cell and trick that host cell into reproducing them. Here's a virus, and what it does is it actually slips inside the host cell, like you see here. And once inside there, it essentially takes command and orders the cell, so to speak, to make copies of itself. And eventually the host cell gets so filled up with the offspring viruses that the host cell itself is destroyed, and the newly made viruses then escape and infect surrounding cells, neighboring cells, and that's how the infection spreads around inside the body. And each of those newly infected cells starts reproducing the virus, and then the infection spreads. And so that's why when I was defining viruses here, I said that they are infectious particles. So that's the only way they can survive is by infecting host cells. Now, here's the structure of a virus. But remember, no organelles, no active enzymes. So what are they made out of? Well, basically, they're just a small chromosome with a protein coat around it, a protective protein coat. The chromosome can be RNA, or in some viruses, it's made out of DNA. It kind of depends on the type of virus you're talking about. And it can be double-stranded or single-stranded. In some cases, it's double-stranded DNA. In some cases, it's just single-stranded DNA. In some cases, it's single-stranded RNA. And in some cases, it can even be double-stranded RNA. And that's kind of unusual because inside of ourselves, matter of fact, inside all other organisms, the chromosomes are always made out of double-stranded DNA. But for the virus, you find quite a bit of variability in what their chromosome is made out of. And the chromosomes are pretty small. Because viruses are so simple, they don't need that many genes. A typical virus might have somewhere between 10 and 100 genes. That's all it needs. Compare that to eukaryotes like ourselves, 25,000 genes. And even simple prokaryotes need around 3,000 genes. All right, and the protein coat that surrounds and protects the chromosome is called the capsid. Oh, what do these genes encode? Well, just the bare minimum that the virus needs to replicate itself inside the host cell. And so some of these genes, for instance, will be for capsid proteins. The virus needs to carry those genes with it. Now, in some viruses, not all viruses, but in some viruses, there's a membrane called the envelope that surrounds the capsid. But the viruses don't even necessarily make the envelope themselves. Sometimes they just steal some of the membrane of the host cell as they're exiting the host cell to make their envelope. Okay, let's watch this virus infect a host cell to see how it gets inside. So the virus has to get inside so that the host cell can start reproducing the virus. But of course, the membrane of the cell is supposed to be a barrier. Amongst other things, the cell is trying to protect itself from viruses, and the membrane is the protective barrier. So how does the virus breach that barrier? Well, the virus keys in on proteins that are in the membrane of the host cell. All cells are going to have some sort of proteins in their membrane. All cells need, for instance, receptor proteins so they can detect molecules outside the cell. They'll need transport proteins for moving things through the membrane, that sort of thing. And all these membrane proteins are always going to have a binding site where they bind to whatever molecule they're supposed to be detecting or supposed to be transporting, well, the virus essentially fools those membrane proteins. Some of the capsid proteins on the surface of the virus are shaped similar enough to the natural ligand of the receptor that they'll bind to the receptor. So the cell will think that it's bound onto a molecule that it's supposed to be detecting, or the cell will think that it's bound onto a molecule that it's supposed to be transporting, when of course it's actually just the virus fooling it. So there's the virus, and click some of those proteins. I didn't illustrate it very well, but some of those proteins look similar in shape to the ligand, so that they'll actually stick to the receptor's binding site, or the transport protein's binding site. And so the virus's capsid proteins share some of that lock and key specificity that we associate with proteins binding to their ligands. The virus then cannot infect cells that don't have the right protein that will fit its capsid proteins. In other words, this requirement that the virus proteins fit into a cell membrane receptor or cell membrane protein of some sort means that this virus can't just bind onto any old cell. It's got to bind onto a cell that has a protein that will fit it with the lock and key specificity. And so that tends to limit the host range of viruses. They can't just infect any old cell type, only certain cell types that have the right protein for them to bind to. And in some cases, this means that viruses can only infect certain species and not other species. To give you some examples, the disease measles and the disease polio only infect human beings. You can't get measles from a horse, or you can't give your measles to a horse, and you can't get polio from a monkey or a goldfish or something like that. The viruses that cause those diseases only infect human beings, as only human beings have the right protein for them to bind to. And even within one species, the virus may be limited into what sort of tissues within that species it can infect, because different tissues within your body express different membrane proteins. Like the virus that causes the common cold, which is called the rhinovirus, binds to some sort of protein that's only expressed in cells in your respiratory tract, like in your trachea and your bronchi and in your mouth and nose. Those proteins aren't expressed in other places in your body, so that virus is limited to just those particular tissues. HIV, which is the virus that causes AIDS, only binds to a certain protein that's found in certain of your white blood cells. So as a general rule, viruses have limited host range specificity. There are some exceptions. There's this virus called the West Nile virus you might have heard about, and it has a very broad range. It can infect cells in mosquitoes and human beings, for instance. And so that virus is keying in on a protein that's found across many different species. That's why it has a very broad range, but that's the exception rather than the rule. Okay, well, to replicate itself inside the host cell, what has to really get inside is the chromosome, because that's where the genes are. That's where the instructions are for making new copies of the virus. How does it get inside? Well, in some cases, the capsid stays on the outside, but the chromosome is passed within, into the cell. And in that type of virus, there's usually a tubular extension from the capsid, so that as soon as all the virus has to do, the capsid has to do, is bind to the receptor, the membrane protein on the host cell, and then this extension of the capsid takes care of the rest, and in goes the chromosome. In other cases, when the virus binds to the membrane protein in the host cell, it triggers the membrane protein to do endocytosis. Remember, endocytosis is when the membrane folds inward and things from the outside of the cell can be brought inside. Well, that process of endocytosis is triggered typically by some sort of ligand binding to a receptor on the surface of the cell. That's what the virus does. It keys in on those receptors, and so the cell starts the endocytosis process, which brings the virus inside the cell, like you see. So one way or another, the chromosome gets inside, either directly or inside the capsid. There's the diagram of endocytosis, and here you see the virus getting inside the cell, inside the vesicle that forms. All right, once the virus gets inside the cell, the chromosome is going to pop out of the capsid, and then it's going to try to get its genes expressed, so this host will make copies of the virus, like that. Now, once the virus has entered the cell, there are two styles of reproduction it can go through. One's called the lytic cycle, and one's called the lysogenic cycle. And just to summarize them in briefly, the lytic cycle is one where the virus gets reproduced as quickly as it can inside the cell. And so what happens is the cell very quickly fills up with the virus, and then the cell bursts like an overinflated water balloon, and it kills off the cell, and the virus spreads very rapidly. In the lysogenic cycle, it's almost like the virus hibernates for a while inside the host cell. After what's called a latency period, where the virus is hibernating, it could be months, it could be years, then the virus will suddenly start reproducing itself and burst forth out of the cell. But let's start with this lytic cycle one, which, as I said, is the one where the virus gets reproduced very quickly inside the cell. And to see how the lytic cycle operates, we need to differentiate between viruses that have a DNA chromosome versus viruses that have an RNA chromosome, because the actual methods that the viruses use to go through the lytic cycle are slightly different depending on which type of virus it is, whether it's a DNA or an RNA virus. And the DNA viruses are a little bit more straightforward, I guess, because since their chromosome is DNA, that's the same type of chromosome that the host cell has, and so it's closer to what the host cell is expecting as far as receiving genes and expressing genes. So the virus doesn't have to bring in any extra enzymes to help it reproduce. Now, as I go through how the viruses go through the lytic cycle, I'm going to be making a big table, and I'm not going to have time to complete the table today, but I want to at least start it here. And so it's going to be a table of enzymes for viral reproduction. And in this table, I'm going to list all the enzymes that are needed for a virus to go through the lytic cycle. But some of these enzymes are going to be host enzymes, where the host cell provides the enzymes. And other of these enzymes are going to be viral enzymes, where the virus has to bring in enzymes because the host doesn't have the right ones to reproduce the virus. All right, so if it is a DNA virus, one of the things that the virus needs to do is express its genes. For instance, like the genes for making new capsid proteins. Those have to be expressed so new capsid proteins can be made. Well, those genes are found on the viral chromosome, just like our genes are found on our chromosomes. And those viral genes have promoters that the transcription factors can bind to. They look for all the world just like the regular old genes in the host cell. As a matter of fact, that's important because as far as the host is concerned, If this just looks like a little chromosome, then the host is going to assume it's one of its own chromosomes and will start expressing those genes. And so, like for our genes to express, we need our RNA polymerase enzyme. We use RNA polymerase 2, but bacteria, for instance, just use RNA polymerase. And the RNA polymerase, of course, uses as its template the template strand in the DNA. And what's the, I know I'm kind of running short on time, but one last thing. What's the product of RNA polymerase when it goes along a template strand? What does it make? Well, in your own cells, when RNA polymerase 2 goes along a template strand, what is produced? Nobody knows what RNA polymerase does? Give you a hint. It's something that gets processed before it leaves the nucleus. Oh, you guys aren't paying attention. Well, I'm not going to tell you. Look it up in your notes. It's something that RNA polymerase makes inside your own cells. All right, study up because you'll need to know it by Friday, but I will see you in Wednesday, or I'll see you today in laboratory.